name is Catherine Matthews. Thank you for watching my video on D2L. I'm going to be talking to you today about antibiotic resistance in agricultural land that has been treated with manure. As most of you already know, antibiotic use is common in livestock industries. Their uses include growth performance, increased feed efficiency, and prevention of disease. About 80% of all antibiotics produced are used for animal industries. Cattle and swine are often fed subtherapeutic doses of these antibiotics in their feed. Some efforts have been seen by the FDA to reduce antibiotic use in poultry, cattle, and swine, especially poultry. So how does all of this relate to agriculture? It is said that the usage of antibiotics in animal husbandry has promoted the development and abundance of antibiotic resistance in farm environments. Manure has become a reservoir of resistant bacteria and antibiotic compounds, and its application to agricultural soils is assumed to significantly increase antibiotic resistance, genes, and selection of resistant bacteria. So what is antibiotic resistance? Antibiotic resistance comes about in a multitude of ways. Mechanisms can include reductases, ABC transporters, acetyltransferases, and even inhibition of protein synthesis. All of this means, in layman terms, is the bacteria develop a mechanism that they can destroy the antibiotic upon entry into the cell, or even defend against its entry to begin with. A lot of the antibiotic-resistant genes can be identified using metagenomic screening of soil in large data sets over a long period of time to notice any relative increases. Sequencing is often done to identify specific genes, and often researchers will use large databases such as GenBank. So my question to you today is there a long-term increase in antibiotic resistance residues in soil treated with animal manure, particularly in cattle and swine? So currently, I am a researcher at OSU looking at the long-term effects of antibiotic resistance in lands treated with cattle and swine manure. I'm looking at a 15-year time period, so I have soil from 15 years ago to today. I have plots from cattle manure, swine manure, and a control plot that was not treated with anything. The antibiotics, some of the antibiotics I've been testing for include the common ones like tetracycline, chloramphenicol, novobiocin, and aminoglycosides. So the big picture of this research, and the middle block is what I'm doing. I am detecting the antibiotic-resistant genes using PCR and culture-based methods. The other two will be developed after the study has been underway for a while. Breaking that down into a simpler set of steps, I take the soil, extract its DNA, use both endpoint PCR and real-time PCR, and then from there sequence to make sure the genes I've analyzed are the right ones. So the results of this study, I tested for a battery of 18 genes. I only detected 4 genes in all 3 of the samples, and the prevalence was relatively constant among them. This suggests that there's naturally occurring resistance in the control environment. From this table, you can see that, again, the results are fairly constant among control, cattle, and swine. However, I must provide a disclaimer here with this is only the pilot study, and a long time period was not used for this initial pilot study. Looking at some currently published research, one article titled Antibiotic-Resistant Genes Spread Due to Manure Application on Agricultural Fields, they used soil treated from a variety of locations, including China, Germany, Austria, the USA, and Denmark. They tested for similar antibiotics, such as the tetracyclines, the sulfonamides, tylosin A, and chlorotetracycline. The results of this study ranked tetracycline as the highest in all soil samples from all of the countries. It increased the most and was found the most prevalent. they found residues of TET-M, TET-O, TET-Q, and TET-W. All of those shortened names mean that those are specific genes a bacteria can harbor and will develop resistance if they have one or two or all of those genes. This study also found that about 30 to 90% of antibiotics fed are not absorbed by the animal and deposited in the waste. Again, this accumulates into the reservoir of antibiotics found in soil. They found that quantification by real-time PCR revealed a transient effect of antibiotic-free manure and a long-term effect of sulfodiazine containing manure on the absolute and relative abundance of the sulfonamide resistance gene SLU-I. All of this means is that they did find an increase in resistance when the soil was treated with antibiotics or with manure that had been antibiotics in the feed. Looking at another currently published research article titled Evidence of Increasing Antibiotic Resistance, Gene Abundance in Archive Soils Since 1940, this testing began in 1940 because penicillin was introduced in the 1920s, and in 1940 it began its mass production. So that's why they chose the year 1940 to begin their study. This study was conducted in the Netherlands, and they had soil that was treated with inorganic manure, organic manure, and mineral phosphate fertilizer. I like this study and have used it in my own study because they have used DNA extraction, quantitative PCR, and sequencing just like I have in my lab. The results of this study say again the tetracycline showed the highest increase in resistance and prevalence. This includes, again, the same genes that were found in the previous study and the study that I've been conducting, TET-M, TET-O, and TET-Q. They also found a dramatic increase in the beta-lactamases and a slight increase in the erythromycins over their study. Most genes were transferred horizontally or via plasmids. That was one of their other discoveries in this research, and that doesn't surprise most of us in the field since there are so many bacteria in soil because it's rich in microorganisms that horizontal transfer is very common of genetic elements. So, closing thoughts. This was taken from the previous study. They said the Standing Medical Advisory Committee's report, The Path of Least Resistance, stated, there's an uneasy sense that microorganisms are getting ahead and that therapeutic options are narrowing. And our results hint that increasing resistance in soil bacteria may be another factor impacting our battle against global AR or antibiotic resistance. So, some questions still remain. The jury's still out. My study states that there's a relative constant resistance even in the natural environment, but other studies prove that there's an increase. So questions that we can still ask ourselves are, should livestock industries stop the use of antibiotics? Do the benefits outweigh the detriment of having antibiotics in the soil? How does it affect the clinical setting? Are any of these antibiotic-resistant bacteria finding their way into our own lives as humans? And are there serious ecological impacts from antibiotic deposits in soil? How is it affecting our natural environment, especially water? In order for us to answer all of these questions, more studies need to be conducted before a definite answer can be made. Again, thank you for watching my presentation on D2L, and if you have any questions, please post them to the discussions page.